Okay, so our last group of bacteria in this lecture series is going to be our Enterobacteriaceae, and these are going to be the largest family of which has many clinically important bacteria. And so here is our laundry list of gram-negative rods that are human pathogens. And so I put next to the ones that are common. Now some of these common ones are things that we don't typically think about, some we do. So E. coli is going to, of course, be common. We have Salmonella typhi, which will be common. Salmonella enteritica is not common. And we have a series of other salmonellas that are not common. Shigella is not common. Yersinia pestis is going to be common. Yersinia enterocolitica, pseudotuberculosis is going to be common. Our Klebsiellas are going to be common. Our Enterobacters, Serratia is going to be common. So again, a lot of these are going to be associated with diseases, and many of them are going to be associated with gastrointestinal diseases, for which you just have to give supportive care for many of these. Now, one thing to keep in mind is that a lot of these that we just talked about are normal flora. And so, but some of these are always going to be causing disease. So, for example, these three over here, Yersinia pestis, Shigella, Salmonella typhi, you do not get colonized with them. They are not part of the normal flora. If they are present in a clinical sample, it is disease causing. Now, on the other hand, if you have in the isolation of E. coli, Klebsiella, or Proteus species, these are normal flora. So you have to do a little bit more with respect to figuring out if they are causing the disease or if this is just part of your normal flora. And typically these guys are going to cause opportunistic infections, meaning that they gain access to some area they're not supposed to be in, or the person's in some sort of compromised state, which allows them to become infectious. Now, these guys are all going to be non-spore-forming gram-negative rods. So here's a picture of our gram-negative rods. They are all going to share a common antigen. It's the enterobacterial common antigen, which is also that LPS core antigen. We'll talk about LPS in just a moment. So LPS has the three antigens, and one of them is a core, and that core is shared across this family. They're going to be fast-growing facultative anaerobes, so they can grow both in aerobic and anaerobic environments. You are going to have them on a non-select bud agar, and then we can use other selective agars such as McConkey agar. The absence of cytochrome oxidase is going to be a fast diagnostic test that can tell the difference between E. coli and other bacterium such as Vibrio and Pseudomonas. So the structure is a gram-negative bacterium. It has LPS on its outer surface. So LPS is that endotoxin. Three components of LPS are: one, it has an O polysaccharide, and this gives it its strains, E. coli O157, that's the O polysaccharide. It has that core antigen that I just mentioned, and that core antigen is going to be found in all of these Enterobacteriaceae. And it also has lipid A, which is the actual endotoxin. So lipid A is the endotoxin portion. They are going to have capsules. So capsules are going to make them more virulent, and those are K antigens. And then they also have flagella, which are H antigens. Now, McConkey agar is going to be able to differentiate these bacterium because it has lactose. And there are some lactose positive bacterium, and then there are lactose negative bacterium. So if you're looking for food poisoning, for example, you can do a McConkey agar and you can grow E. coli, or there can be no growth, so it might lead to a Salmonella or Shigella type of inquiry. But basically, on McConkey agar, your lactose-positive bacteria are going to look either pink or purple. Another thing are biosalts. So biosalts are made by your liver. Well, they're made by your liver, put into your gallbladder, stored, and then released when you eat. And so biosalts are going to be, your normal flora will be sensitive to biosalts, but your pathogenic bacteria that are gram-negative rods are resistant to biosalts. And so if you have a agar with biosalts, it's going to be selective for gram-negative bacteria because they're very potent against gram-positive bacteria. Now, pathogenesis of this group are going to be, again, our endotoxin, just like any gram-negative LPS. They also are going to have capsules, or the species that are more pathogenic will have capsules, and this is going to protect them from phagocytosis, from complement, and from antibody. If you do have anti-capsule antibodies, they will be protective. They have an antigenic phase variation, and what this means is those antigens we just talked about, your flagella, your capsule, these can be expressed or not expressed. The bacterium can actually make a choice. So, for example, if a bacterium, you have anti-capsule antibodies, but the bacterium decides I'm not going to make a capsule, then those anti-capsule antibodies will not help to protect against that particular bacterium. They also have type 3 secretion systems, which are a molecular syringe, and these are going to, the bacteria are going to inject things into, especially macrophages. And some things they can inject are things that can prevent phagocytosis, for example. They also are very good at getting nutrients. They love to get iron. They are going to compete with our iron chelating compounds, so they have their own chelating compounds. They have siderophores, which will go around and bind up free iron. They also have enterobactin or aerobactin, which are chelating compounds to grab iron. Now, E. coli is going to cause gastroenteritis, and this is the percent of bacterium that actually also go on to form bacteremia, get into the blood. And you can see E. coli is about 45% of the bacteremia. There are different types of E. coli. We have enterotoxigenic E. coli, pathogenic E. coli, and aggregative E. coli, and they're all going to cause infections in the small intestine. And then you have your more invasive Shiga toxin-producing E. coli or enteroinvasive E. coli, and they are going to be having disease in the large intestine. So I'll just go through each of these really briefly and talk about some things that are important to note. So enterotoxigenic E. coli, again, is in the small intestine. This is often referred to as traveler's diarrhea. And so this is fecal material contaminating food, so fecal oral route of contamination. This is going to infect the small intestine, and you're not going to have any bloody diarrhea. So it's mostly going to be watery diarrhea with cramps. They do have heat stable toxin, STA and STB. STA is in the human disease. They have heat labile toxin, LT1 and LT2. Heat stable basically means if you heat this up, it will not break down. Heat labile means if you heat it up, it will break down. So the STA is what's going to increase cyclic GMP, and this is going to lead to hypersecretion of fluid, so that leads to the diarrhea, and inhibition of fluid absorption, so you get dehydrated. LT1 is going to be similar to cholera toxin. It leads to cyclic AMP increase, and that's going to lead to enhanced chloride secretion and a decreased absorption of sodium chloride, which is, of course, a major osmoregulator. Enteropathogenic E. coli, again, is in the small intestine, and you're going to have this associated more with an infant diarrhea. It's very rare in the United States. It's going to, again, have watery, non-bloody diarrhea, but this time it will have vomiting. In the case of the pathogenic, you have pathogenicity islands that are called the locus of enterocyte effacement. And this particular bacterium has attachment effacement properties. And what it basically does is it attaches to epithelial cells and destroys the microvilli. So over here, if you look at this image, you can see these balls, and these are the microvilli being um, effaced, being kicked off. And then all these little bacterium are invading, or they're inside some sort of cell vesicle. And so attachment effacement is a property of enteropathogenic E. coli. Okay, and then we have enteroaggregative E. coli. An enteroaggregative E. coli, again, is going to be associated more with infant diarrhea, traveler's diarrhea, watery, non-bloody, with vomiting and low-grade fever. This guy does an autoagglutination, which makes a stacked brick arrangement. So if you look at this image, this is an H&E stain, and you can see that they kind of look like this little wall of bricks, so that was given the term stacked bricks. Now in the large intestine, you have your bloody diarrhea. So we start with our Shiga toxin like E. coli, and this one is going to initially have a watery diarrhea progressing to blood, hemorrhagic colitis, and this will have cramps without fever. This can also lead to eventually hemolytic uremic syndrome, which can be acute renal failure, thrombocytopenia, and a microangiopathic hemolytic anemia. It has toxin A, which is going to cleave the 28S ribosomal RNA, so it blocks protein synthesis. This is similar to Shiga toxin. And then you're going to have some are going to have that attachment effacement genes that we saw previously with our pathogenic E. coli. Now, even though O157H7 is the most common cause of human disease, there are many others that can cause disease. It was originally that they would look for the O antigen and H antigen to diagnose the Shiga toxin like E. coli's. But now they just simply look for the presence of Shiga toxin. And another thing to point out about this E. coli is that you only need a small amount of bacteria to actually infect. In some cases, like Salmonella, you'll need upwards of 800 bacteria to infect. But for your Shiga toxin E. coli, you only need 100. Now, enteroinvasive E. coli is going to be very rare in all countries, and it's going to infect the large intestine. Not much to say about this one. You're going to have watery diarrhea, progresses to dysentery with bloody stools. And the invasive part of what causes the bloody stools, it doesn't have a toxin, but it's able to invade, it's able to destroy the epithelial layer, it causes leakage, and you get blood leaking into the large intestine. Now, one thing to remember about E. coli is that it can cause other infections as well. One thing that's important is neonatal meningitis. E. coli and group B strep are going to be the most common cause of meningitis in children very young. And also you can get septicemia, which is basically blood poisoning. So the infections that are going to lead to septicemia are usually going to start in the gut or urinary tract. And mortality is very high in immunocompromised patients with respect to septicemia. Now, a related bacteria is Salmonella, and so Salmonella is going to be invading your M cells, and so this is in your gut, of course. Salmonella also can enter your epithelial cells. They like to transverse across and get into your body. And then once inside your body, they can get picked up by dendritic cells, macrophages, neutrophils, which can try to kill them. Now, Salmonella does not replicate in the cytosol. Salmonella is similar to Mycobacterium in that it replicates inside of a vesicle. Now, they have pathogenicity islands, and they have type 3 secretion systems as well. Most are going to be infections from poultry, eggs, or dairy. These Salmonella is a normal flora bacterium in these domesticated animals. It's going to lead to gastroenteritis, which is going to be a non-bloody diarrhea. and then you're going to get vomiting and cramps after consumption. Septicemia is going to be higher in pediatric and geriatric. And you also have enteric fever. So salmonella causes enteric fever, which is typhoid fever. So basically what's happening here is you're getting your salmonella in the same way as described here, but then you get salmonella in, let's say, a macrophage, and here's your salmonella in the vesicle, and the macrophage takes a hike and travels to the liver or other tissues. And when salmonella gets to the liver, of course, makes bile and biosalts and sends everything into the gallbladder. And also you're moving things around systemically, so you're going to get flu-like symptoms, malaise, and salmonella can be released back into the gut through your bile. And also, Typhoid Mary was famous because she had a chronic infection in her gallbladder. And so she would constantly shed salmonella, and she was a cook for families. And so through fecal oral contamination, she would spread salmonella typhi to people that she cooked for. Now, Shigella is also going to be a gram-negative rod. It has replication in your large intestine, your colon. It's going to have type 3 secretion systems that secrete 4 proteins, IPA, A, B, C, and D. These are going to induce membrane ruffling, which initiates uptake of the bacteria into your colon epithelial cells. These guys are going to survive in the cytoplasm and replicate in the cytoplasm. They can rearrange actin to propel themselves to the next cell, so you get cell-to-cell passage. Shigella dysentery is going to have the Shigella toxin, and this again, like E. coli, is going to cleave that 28S RNA, block protein synthesis. For Shigella, humans are the only reservoir. There is no animal carrier, and pediatric diseases are going to be the most of the cases with Shigella. And then we have Yersinia pestis, which is plague, and it's very fatal. Yersinia pestis has the ability to resist phagocytes, so it's going to have a type 3 secretion system that transfer proteins and prevent phagocytosis. It has 2 plasmids. One will have a capsule. One will have something called the plasminogen activator protease, and this is going to degrade complement. It actually targets C5 and C3 specifically. And so this enables it to evade the innate response, and fibrin clots can be degraded as well so that you can get dissemination. Now, Yersinia pestis is a zoonotic infection, meaning that it infects an animal rather than us. So we are an accidental host. We have urban plague, in which rats are the host. Salvic plague, in which you have more wild animals, but also domestic cats can be the host. Fleas are going to be the vector. With respect to bubonic plague, you get a bubo, which is basically a swelling, and bubonic plague is 75% lethal if it's not treated. Pneumonic plague is going to be the one in the lungs, and you can get person-to-person spread with this, and this is 90% lethal if not treated. Other gram-negative rods that are important are Klebsiella and Proteus species. Klebsiella are going to cause an atypical pneumonia with blood-tinged sputum. Atypical is going to be for any pneumonia not caused by staph or strep. Proteus species are going to infect the urinary tract. They're going to cause cystitis, and if they get to the bladder, and they can also disseminate into the kidney. They produce urease, which splits urea into carbon dioxide and ammonia. And what this does is raises the pH of the urine, and so you get crystals that form because you're going to get precipitates of magnesium and calcium called strivate and apatate. And these are going to be, of course, kidney stones.